All right, immunology is cool, but there's kind of a lot of stuff to go over. So I'm not sure how comprehensive this is going to be, but hopefully I'll make a couple of these, and there'll be like some nice visual summaries to kind of get you started, I hope. So BUGGED episode 1, we'll be talking about innate versus adaptive immunity, and also the complement system. And again, use your notes to fill in the details. So when you think of the immune system, you probably think of that kind of long-term memory, like antibodies and all that. But there's also obviously the short-term, immediate acting thing as well. So we can divide our immune system into the first guys, which is the innate responders, and then the second guys, which come later on, and that's the long-term memory guys, which is your adaptive immune response. Obviously, before that, you've obviously got your physical barriers like your skin and your mucus and your stomach acid and all those nice things, and like lacrimal glands and all that, tear ducts. So adaptive immunity is your long-term specific one, and these are the ones that kind of ultra specific. So for example, COVID vaccine, you've now got antibodies against COVID, hopefully, in your bloodstream, and those are obviously very specific to COVID. And so this is long-term memory. And the way I remember long-term is lymphoid pathway. So these cells, there's basically B and T cells, and they come from the lymphoid pathway. B and T cells are specific because they have different receptors. So the receptor on the B cell, you probably know it's called the antibody, and the receptor on the T cell is just called the T cell receptor, so it's not that creative. Also, why is it called an antibody? Why is the B cell receptor called an antibody? I thought the B cell makes antibodies. Well, essentially what happens is on your B cell, you have an antibody which acts as a receptor, and that's kind of very specific, just kind of by chance, to an antigen. When an antigen binds into that antibody, loads of those antibodies will be made, so that's kind of monoclonal antibodies. And, sorry if I'm daydreaming randomly, but when loads of those antibodies are made, they are no longer glued to the surface. They're free to kind of go and circulate in the bloodstream. So those antibodies that were initially the receptors go and transform into circulating antibodies. That is called the humoral response. Humor meaning blood. But yeah, okay, we'll talk about that later in another video, maybe. So obviously B cells and T cell receptors are very specific. How do they generate that specificity? Again, heads up for later on, so you don't need to know this just now. But you know how proteins are made of genes? So basically through some, for example, you have a gene with the codons ABC that is going to lead to a nice protein. If you have some genetic magic that rearranges the codon slightly, for example, instead of ABC to ACB instead, you're going to end up with a slightly different protein. So essentially, this happens in B and T cells, and it's called VDJ recombination. And this leads to these antibodies and the T cell receptors having different kind of specific shapes. There's also ways of introducing mutations later on, etc., to make these things mutate. But to make these things specific. Okay, don't worry about those for now. Just remember that B cells have the receptor called the antibody, and T cells have the receptor called the T cell receptor. So our innate immune system is the other one. That's your first line of defense. That's short-term, rapid-acting, and non-specific. So you're kind of thinking of the stuff that does like phagocytosis and the kind of first-line patrolling things. And these come from the myeloid pathway. And so one cell is called the neutrophil, and neutrophils have like lots of nucleuses in them. So they're also called PMN, which stands for polymorphonuclear cell, I think, basically. Essentially, it has lots of nucleuses in it. So you can recognize that distinct shape under the microscope. There's macrophages, which are obviously our first-line gobblers. Neutrophils are more like gobblers, but kind of primed more towards bacteria. And then dendritic cells, they're the snitches. Essentially, what they do is they take up antigens from bacteria and whatever, and they go along and they migrate to B cells and to T cells, and they're like, yo, let's come, there's a problem here. So these guys snitch on the pathogens, essentially. You come across dendritic cells later on as well when we're talking about like skin immunity or whatever. Basically, they kind of lie low on the surface, they wait for antigens, and then when they detect the antigen, they swim away up into the lymph nodes. The lymph nodes are kind of your, the kind of tissues that contain lots of B and T cells. So the dendritic cell kind of swims up there and then presents the antigen to the B and T cells. So the B and T cells, they're not generally floating around that much. They kind of live in the lymph nodes, and they rely on these guys to activate the adaptive immune response. So the innate immune response can do a couple things, but the first thing it can do is cause inflammation. So remember, like, if you have, for example, if you've been exposed to something before and you have the memory, your immune system knows what to do. It goes in like a pro, and hopefully you should recover quickly. If you have like a virus that you've never encountered before, you can get very sick because basically your immune system doesn't know what to do. It kind of panics and it's like, okay, the only thing I know to do is to basically just have massive inflammation. So inflammation is when you recruit loads of immune cells. And so that's what it does. It basically releases hormones, and these hormones are called cytokines. So immune hormones are called cytokines, specifically interleukin 1, interleukin 6, and also tumor necrosis factor alpha. TNF alpha is also like very important for loads of diseases throughout medicine. So on it, and these interleukins are going to basically cause inflammation. So they're going to essentially lead to proteins. For example, a type of protein called a C-reactive protein, which we'll talk about. But C-reactive protein is a marker of inflammation, essentially. But so these cytokines will lead to your body producing proteins, and these lead to basically you feeling unwell. So inflammation has Inflammation has several signs, right? Like fever, redness, whatever. And there's kind of 5 signs that you need to know. So pain, obviously, heat. So your area, like for example, if you had a cut and it becomes hot and inflamed, it's going to become like red and hot, and so red is another sign. It's going to become swollen due to more, like your blood is going to increase blood flow there to try and get more immune cells there. So it's going to become, you're going to have edema. Edema essentially means swelling. And also, say you have a massive lump on your hand, for example, you're going to have loss of function. So there's Latin terms for those as well, like rubor, whatever, cala, but I don't remember them. Essentially, you need to know 5 signs of inflammation: pain, heat, redness, swelling, and loss of function. And those can be caused by the innate immune system basically being upset. It doesn't know what to do, and it panics, makes these little hormones, and these hormones make proteins which make you upset and give you those nasty symptoms. Okay, so this is one thing that innate immune system can do. And so say we have a bacteria or any pathogen, and that has broken through your initial physical defenses, so through your skin, you've got cut. And your body's not been exposed to this, or your body doesn't know that there's something that's up. What happens is there's guys patrolling, just like old men. They have nothing to do, so they kind of patrol. And these old men, they're called the innate immune cells. So, for example, your neutrophils, your macrophages, your dendritic cells, they're kind of just chilling in the blood until they recognize something. And how do they do this? They recognize this. They have a receptor, which is very non-specific, so unlike the B cell and the T cell receptor, and that is called a pattern recognition receptor. An example of the pattern recognition receptor is called the Like Receptor 4, or just in general Like Receptor, and it's a horseshoe shape, and essentially the antigen kind of fits in it, and it's going to cause problems. So the antigen is called a ligand. So like in ligand in chemistry, ligand is like a bit that kind of just fits onto something. So the antigen is a ligand, and it acts as a pathogen-associated something, but basically a PAMP. That's what you need to know about. You don't need to know about what it stands for, I think. So the antigen is going to come across. We can call the antigen a PAMP now. And it's going to fit into the toll-like receptor in the innate immune cell, which is one of these cells. And this innate immune cell is the first thing is now is recognize that a pathogen has come. The next stage is going to sound the alarm bell and basically get inflammation and get everyone kind of going. So amplification. And our third kind of step in the innate immune response is time to die. So essentially, there's 3 ways that it's going to try and kill this guy. One is phagocytosis, so like munching. Second is extracellular killing, which is complement, which is going to be like what we're going to talk about. And third is the antiviral state. What is the antiviral state? So say this is a virus, not a bacteria. What happens is your cells will produce basically types of proteins which stop replication. So obviously viruses hijack your cell. Your cell is going to have like a defense mechanism, and it's going to activate that with these interferons that are produced in the antiviral state. These will kind of stop viral replication and hopefully make the virus die. So what we're going to talk about is the complement system, which is part of extracellular killing. It's one example. It's the biggest one, and it's kind of complex. But yeah. Okay, so what is complement? Complement is a system of extracellular killing. It can be activated by a couple of things. And essentially, the end product is we're going to have 3 things that are essentially going to destroy pathogens. So complement starts with a protein that's floating around your blood. If you've done like the clotting cascade, it kind of is similar to this. It's another kind of cascade. So you have C3 protein floating around in your blood. And then when it gets activated, for example, by this guy recognizing stuff, a bacteria, for example, it is going to convert by an enzyme called C3 convertase into 2 small bits, the C3A and the C3B, so the 2 shades of green. So essentially this enzyme splits them into 2. And then there's going to be a couple functions. So C3A is going to get married to a guy called C5A. And these guys, what they're going to do is they're going to form, when they're married, they're kind of like a power couple. And this combination can do one. The first thing it can do is it can activate neutrophils, and it basically attracts neutrophils to the area. So once this has happened, loads of neutrophils are going to come, and this is via chemotaxis. So kind of when your neutrophils kind of play it with your bacteria, loads of neutrophils are recruited to your area, which is an example of inflammation. So when you have immune cells coming, that's inflammation. However, if you've ever had like a spot that's got pus in it, for example, that's neutrophils. So when essentially you have too many neutrophils, they can release poisons essentially, and whatever, those can turn into that green nasty pus. So we're seeing here how there's a balance between getting the neutrophils there and getting them to kill stuff in time. But if we get too many, inflammation can have those bad effects as well. So inflammation is not necessarily bad, but it can have bad effects. That is a common theme in immunology. Okay, so that's C3A and C5A. C3B, so our other product of this, he's a sticky guy. So he also has 2 functions. He has 2 functions. One of them, he's quite sticky. So what he's going to do is now he's super sticky like honey, and anything he touches, he's going to stick to. So even if that's a cell or protein or by any chance, if that's a bacteria, he's going to stick to it. And when it sticks to that is basically like a target saying, like alerting all the macrophages that there's a bacteria here. So it's going to stick to something and then prime the bacteria for gobbling by macrophages, and that's called opsonization. So basically acting as an alarm system. And C3b has another function. It's going to activate C5 into C5b. This is separate from the C5a. It's going to activate C5 into C5b. So C5 becomes activated. So this is showing C3b activating C5. Then C5b is going to go along with these guys, C9, C8, C7, and C6. So just 9, 8, 7, 6, and 5b. 9 is probably the most important one. And these guys, they club together and then they kind of batter and hammer anything that comes in their way, well, especially bacteria, and they're going to destroy the membranes. So especially C9, but it's going to work with the help of 8, 7, 6, and also 5b, and that's going to form the membrane attack complex and basically drill a hole in stuff and cause them to bleed to death. Not bleed, but cause the bacteria to kind of leak to death. That is essentially complement in a nutshell. The only complex thing we have to do now is talk about the different ways that you can activate complement. Obviously, the first way that we talked about is through your innate immune system. For example, a toll-like receptor recognizing a bacteria, and that would be called often the alternate pathway. The alternate pathway is when you have something, an innate immune cell, that is going to recognize an antigen on a gram-negative bacteria. What is gram-negative? Essentially, we've stained bacteria in different stains. They can either stain purple, meaning they're gram-positive, or if they don't absorb that purple stain, they stain red, which is called gram-negative. So gram-negative bacteria, and there's other types of bacteria as well that don't stain with either. But gram-negative, they look red under the microscope. That's why I've highlighted it red. They often contain a kind of sugar, kind of glucose thingy called lipopolysaccharide. When that gets detected, that lipopolysaccharide can act as a ligand that can get detected by the toll-like receptor, by the immune system, by the innate immune system, that can activate complement. So that's one way that we can activate complement. You need to know that. The second way you can activate complement is your classic pathway, which is basically through antibodies. So if you think, what do you classically think of when you think of immune system? You think of antibodies. So therefore, the classic pathway is basically antibodies. So an antibody binds to an antigen. It can also activate this kind of killing mechanism. So this is one way that antibodies actually kill, through activating complement. Now you have to know this, before we get to the juicy C3 whatever, the classical pathway can be activated by an antibody, or also this guy, C-reactive protein. So you can either have antibodies, or inflammation can activate the classical pathway, inflammation through C-reactive protein, which is made by your liver, an acute phase protein. And essentially, so what you can do is you can think of it as there's like a pathway that activates the complement pathway, and it goes into CRP, or the antibody, so that will be your first step. And then it goes to a C1 protein called C1q, that turns into C1r, and that turns into C1s, and finally that activates C3 protein, whatever. How do you remember that? Well, it's quite easy, because we go CRP is our first stage, or antibody, but if you think of it as CRP, PQRS, PQRS, CRP, C1q, CR, C1R, C1s, and then that activates 3, and then from there, the rest is the same. So there's one pathway here, the alternate pathway, which doesn't involve these guys. It just goes straight to C3, and then from there, the classical pathway involving antibody or CRP goes to Q, RS, and then this. And the final pathway is called the lectin pathway. So if microbes have certain sugars, which is called lectin, that gets essentially converted into something called NBL, and then your body is going to make something called MASP1, and that is going to activate complement as well. So essentially you have 3 basically precursors that can activate complement. Alternate pathway, which basically goes straight to C3, the classical pathway, which goes via these guys, and the microbial sugar pathway, the lectin pathway, which goes via MASP1 and also activates complement. Hopefully that kind of makes sense.